Okay, we're going to talk here really quickly about how DNA copies itself. That's what DNA replication refers to. Replication means to copy something. So this is happening during interphase. The cell is copying its DNA. It's going from having monovalent chromosomes to having bivalent chromosomes so that the cell can then divide later on. So this is DNA replication, the easy version. Okay, step number one, the DNA untwists. It's using an enzyme called helicase. So that is what causes the DNA molecule, the double helix, to untwist. Then the DNA unzips. There are very weak hydrogen bonds between the A and T and the C and Gs, the nitrogen bases, so they pull apart very easily. There are very strong covalent bonds between the sugar phosphate molecules, so the backbone of the DNA never separates. Okay, next, an enzyme called DNA polymerase reads each side of the DNA and it attaches the correct opposite nucleotide to the complementary base pairs. So for example, the enzyme up at the top, it reads a red nitrogen base, so it attaches a blue nitrogen base. So let's say that's an A and he's attaching a T. Then that enzyme slides down to the yellow one and it reads yellow and it attaches a green. So maybe it's a C and attaches a G. Then it slides down to the last one. It reads a blue, maybe that's a T and it attaches an A. At the same time, on the opposite side, there is a DNA polymerase that is moving upward, up the side of the DNA, and it's copying the red side and adding a blue, it's reading the green side and adding a yellow, it's reading the blue side and adding a red. And if you imagine this in your head, you would have created two identical sides. Okay, as we said, as you imagined, the two copies now, you have two identical copies. Notice the side on the left is identical to the side on the right. What you can't see is that what you've actually created are the two sides of a bivalent chromosome. So you had one chromosome and now you've created, or you had a monovalent chromosome and now you've created bivalent chromosomes. And then in the final step, the DNA twists again to form a double helix. Okay, by the time that the DNA polymerases have traveled all the way through the DNA, they make about one error in every 10,000 paired nucleotides, which seems like not very many errors, although because we have about 3 billion letters, that actually ends up being a lot of errors. Each change is a mutation, and each mutation is capable of leading to some disastrous side effect. So it turns out that our bodies have proofreading enzymes and repair enzymes. Remember back to our cancer discussion where we talked about the guardian angel gene. That's where the guardian angel gene comes in. These proofreading and repair enzymes check for mistakes and fix the mistakes so that by the time they're done, there's only about one mistake for every billion nucleotides. If we have 3 billion nucleotides, it means about 3 mistakes, which is a tiny percentage in the entire cell every time that it divides. And just as an FYI, DNA can also be damaged by carcinogens, like we learned about in the cancer unit, and ultraviolet radiation, which is a kind of carcinogen.